Welcome to the deep dive. We're ready to explore some biophysics.
Absolutely, let's dive in.
Perfect. We've got some university-level biophysics course materials to go through.
Sounds fascinating. Where should we start?
Well, the listeners got notes on atoms, lasers, cells, and more.
Wow, a pretty wide range.
I know, right? It's amazing how these tiny things we can't even see impact our lives so much.
Yeah, definitely.
So maybe we should start with light itself.
Okay, light.
The listeners' notes talk about light having this dual nature. What does that even mean?
Well, it's a concept that basically changed physics as we know it. Scientists always wondered, is light a wave or a particle?
Right, makes sense.
Turns out it's both. It kind of acts like a wave in some situations and a particle in others. It's a pretty crazy idea. But it's been proven over and over again.
So how can something be two things at once? That seems impossible.
Yeah, it's one of the big mysteries, but it's at the core of quantum mechanics. That's the theory that governs how things work at the atomic level. Super tiny scales.
Okay, so quantum mechanics. Got it.
And a really good example of this dual nature is the double slit experiment.
Oh, yeah, I've heard of that. But how does that work again?
Okay, so picture this. You shine a light beam through two tiny slits. Now, if light were only a wave, you'd see a really simple pattern on a screen behind the slits, like ripples spreading out in a pond.
Right, makes sense.
But what you actually see is way more interesting. You get this interference pattern with bright and dark bands. It's as if the waves of light are kind of bumping into each other. That's light acting like a wave.
Okay, yeah, I remember seeing those diagrams in my science classes.
Right. Now here's where it gets wild. Imagine dimming the light way down. So much that only a few photons are going through those slits at a time. Photons are like tiny packets of light energy.
Okay, photons, tiny light packets.
And when you do that, what you see on the screen are these little dots. It's like each photon is a tiny particle hitting the screen.
So each little dot is a photon.
Wow. And the crazy part, it's like each photon goes through both slits at the same time, interfering with itself.
So is it a wave or a particle then? I'm so confused.
It depends on how you look at it. It's as if light decides what it wants to be based on how we're trying to measure it.
That's so strange.
I know, right? It totally changed how we understood physics. And it led to, well, quantum mechanics.
So this dual nature of light, does it affect us in any way? Or is it just something theoretical?
Oh, it totally has real-world effects. I mean, think about lasers. They wouldn't exist without understanding this wave-particle duality.
Wow, okay, so it's not just some abstract physics concept then.
Not at all. Lasers are used everywhere, from scanning groceries to super precise eye surgeries.
That's pretty amazing. Okay, so let's move on to the atom itself, right? How does all this change how we think about atoms?
Well, it means we got to rethink that old picture of electrons orbiting the nucleus like planets around the sun.
Right, I remember that from school.
Instead, we have to imagine electrons existing in these kind of blurry, cloud-like areas called orbitals.
Orbitals. Okay, I'm already getting a little lost. Can you explain what you mean by orbitals?
Sure. So first, imagine the atom having different energy levels, kind of like steps on a staircase. Electrons can be on these different steps, these energy levels, and the lowest level, that's called the ground state.
So electrons can jump between these energy levels.
Exactly. If an atom absorbs some energy, an electron can jump up to a higher level, like climbing the stairs. And when an electron falls back down, it releases that energy, often as light.
Okay, that makes sense. But where do these orbitals fit in?
An orbital basically tells you the probability of finding an electron at a particular spot around the nucleus. It's like a 3D map of where the electron is most likely hanging out.
So it's not a fixed orbit like a planet.
Nope. It's more like a cloud of possibilities. And these orbitals come in different shapes depending on the energy level and other factors.
Oh, right, like this orbital and the polar orbital. I remember those diagrams, kind of.
You got it. The Acer orbital is spherical and the porbital looks like a dumbbell.
Right, but why are there different shapes? It seems kind of random.
The shapes actually depend on the wave-like behavior of electrons, and they're described by something called quantum numbers.
Quantum numbers. That rings a bell, but I'm a little fuzzy on the details. Can you remind me?
Sure. Think of quantum numbers as an electron's address inside the atom. Each electron has its own unique set of 4 quantum numbers. And these numbers tell you the electron's energy level, the shape of its orbital, the direction it's pointing in space, and even its spin.
Okay, so it's like a code that tells you everything about an electron in the atom.
Exactly. And these quantum numbers follow certain rules, like the Pauli exclusion principle, which says no two electrons in an atom can have the same set of those four numbers.
So each electron needs its own unique address.
Right. It's like a cosmic game of musical chairs. And this principle helps us understand why we have all the different elements in the periodic table and why they have their unique properties.
That's incredible. So it's like these abstract rules about quantum numbers actually shape the entire world around us.
Yeah, pretty much.
But wait, my notes also mention something called the Stern-Gerlach and Einstein-de-Haas experiments. What are those about?
Those experiments showed us another really cool thing about electrons. They have spin.
Spin, like they're spinning around.
Exactly. Imagine an electron spinning like a top. This creates a tiny magnetic field.
So not only are electrons in these weird orbitals, they're also spinning.
That's right. And that spin is super important for things like magnetism.
This is all so mind-blowing. But how do we actually study these tiny particles and their properties?
That's where tools like lasers and spectroscopy come in. They're like our windows into the microscopic world.
Lasers, now we're talking. They seem to be everywhere these days, from supermarket scanners to cutting-edge medical devices. How do they actually work, though?
Well, a laser relies on a few key ideas: spontaneous emission, stimulated emission, and what we call population inversion. Imagine an atom, right? It has these energy levels, kind of like rungs on a ladder.
Yeah.
Usually most atoms are chilling on the bottom rung, their ground state. Right, like we were talking about before.
Exactly. But if you zap them with energy, electrons can jump up to higher rungs, those excited states.
Okay, I get that part.
Now, when an electron falls back down a rung, it releases energy, often as light. That's called spontaneous emission. But here's the cool part about lasers. If you hit an excited atom with a photon that has just the right energy.
A photon being a tiny packet of light.
Yep. That excited atom can be stimulated to release a second photon, and the second photon is identical to the first one.
So it's like one photon triggers another one.
Exactly. It's like a chain reaction. And to make this happen efficiently, you need more atoms in an excited state than in the ground state. That's called population inversion.
So you've got to pump them up to those higher energy levels.
Right, and that creates an environment where stimulated emission can take over. And that's what gives you that really intense focused beam of light we call a laser.
Okay, that makes sense.
No.
The notes mention the ruby laser specifically.
Ah, yes. The ruby laser was one of the first lasers ever built. It uses a synthetic ruby crystal as the medium that produces the laser light. And to pump those chromium atoms in the ruby up to an excited state, it uses a flash lamp.
So that flash lamp is providing the energy to create that population inversion.
Exactly.
Fascinating. So we've got these lasers. Now what? How do we actually use them to study tiny stuff? The notes mention something called absorption spectroscopy.
Absorption spectroscopy is all about shining light through a sample and seeing how much of that light gets absorbed at different wavelengths.
Wavelengths being like different colors of light.
You got it. It's kind of like shining a flashlight through colored water. Some colors pass through, some get absorbed by the molecules in the water. By looking at which wavelengths get absorbed, we can identify the molecules in the sample and even figure out their structure.
So it's like using light to create a fingerprint for different molecules.
Hmm.
I remember the notes mentioned something called the Jablonski diagram too.
Ah, yes. The Jablonski diagram is just a way to visually represent the different energy levels a molecule can have and how it transitions between them when it absorbs or emits light.
So it helps us visualize what's happening at the quantum level. And what about this Lambert-Beer law? What does that tell us?
The Lambert-Beer law gives us a mathematical connection between how much light is absorbed, the concentration of the molecule we're looking at, and the path the light travels through the sample.
So if we know two of those things, we can figure out the third, like maybe the concentration of a substance.
Exactly. And that principle is used in all sorts of applications. For example, a lot of medical tests use the Lambert-Beer law to measure things in your blood.
Wow, so this is all very practical stuff.
Absolutely. By simply shining light through something and seeing how it's absorbed, we can learn a ton of information. But the notes also talk about something called fluorescence spectroscopy.
Right, fluorescence. How is that different from absorption spectroscopy?
While absorption spectroscopy looks at what wavelengths of light are absorbed, fluorescence spectroscopy focuses on the light that is emitted. Some molecules, when you hit them with light of a specific wavelength, will emit light at a longer wavelength.
So it's like they absorb one color of light and then glow with a different color.
That's a great way to put it. And we can use this fluorescence to study all sorts of things, like how molecules interact or what's going on inside cells.
The Jablonski diagram is useful here too, right?
Absolutely. It helps us see the energy transitions that occur during fluorescence. And there's this thing called the Kasha rule, which basically says that fluorescence usually happens from the lowest excited state. That's why the emitted light always has a longer wavelength than the light that was absorbed.
That shift in wavelength, I think it's called the Stokes shift, right?
Yep, you got it. And by looking at the Stokes shift and other things about the emitted light, we can learn about the structure and behavior of molecules.
This is all so cool.
Hmm.
It's amazing how we can use light to study the microscopic world.
It really is remarkable. But before we get too deep into the world of light and molecules, maybe we should take a detour and talk about a technique that lets us look at individual cells. It's called flow cytometry.
Flow cytometry, I've definitely heard of that, but I'm not entirely sure how it works. Can you give me the rundown?
Of course. So imagine you have all these cells, right? And they're labeled with these fluorescent tags.
Fluorescent tags, like little glowing markers.
Exactly. And they're all flowing in single file through a laser beam. That's basically flow cytometry.
So it's like each cell gets scanned individually.
You got it. And the light that's scattered and emitted by each cell gives us information about its size, its shape, even what's inside it. We can actually identify different types of cells based on their own unique fluorescent fingerprint.
Wow, that's so cool. What kind of information can you actually get from flow cytometry?
Well, we can figure out how big the cell is, how grainy it is. That can tell us if it's dividing, for example, or if something's changing inside. We can even see how much of certain proteins a cell has, like those on the surface of immune cells or even cancer cells.
So you can actually use flow cytometry to diagnose diseases.
Absolutely. It's a super important tool for diagnosing blood cancers, like leukemia and lymphoma. Doctors can look at the different types of blood cells and how many there are, and spot any abnormalities that might signal a problem.
That's incredible. It's amazing how we can use these techniques to actually look inside the microscopic world and get clues about our health.
Yeah, it really is amazing.
Speaking of looking inside things, the notes mention X-rays next. I definitely know those from going to the doctor's office.
Right. X-rays are another way to see what's hidden, but they work way differently than light or lasers. They're part of the electromagnetic spectrum, just like visible light, but with much higher energy and shorter wavelengths.
So that's how they can go through our bodies and show images of bones and organs, right?
Exactly. X-rays are generated in a vacuum tube, where you have electrons being accelerated towards a metal target. When those electrons hit the target, bam, they release energy as X-rays.
Yeah, I vaguely remember those diagrams from physics class. So how do they actually use X-rays to make those detailed pictures?
Well, there are different ways to use X-rays for imaging. A common one is radiography. It basically involves shooting X-rays through the body onto a detector, creating a shadow image of the stuff inside.
So denser stuff, like bones, absorbs more X-rays, and that's why they show up whiter in the picture.
Exactly. While softer tissues let more X-rays through and appear darker, and by adjusting the strength and how long the X-rays are on, doctors can get images of different parts of the body with different levels of detail.
Okay, that makes sense. The notes also mention X-ray diffraction, though. What's that about?
X-ray diffraction is a bit different. It uses the wave-like nature of X-rays to figure out the structure of crystals. That includes biological molecules like proteins and DNA.
So instead of pictures of our insides, this technique helps us see how atoms are arranged inside those molecules.
Precisely. When X-rays hit a crystal, they scatter off the atoms in a very specific pattern. That pattern, the diffraction pattern, can be analyzed to figure out the 3D structure of the crystal.
Wow, it's amazing how those invisible waves can show us how things are built at such a tiny scale.
It is pretty incredible.
Okay, so moving on, I'm curious about this next topic. Fluid dynamics and circulation. It seems like a jump from X-rays to blood flow.
It might seem like a big change, but the laws of physics apply everywhere, from the tiniest molecule to our whole bodies. Fluid dynamics is basically understanding how fluids behave, things like liquids and gases under different conditions.
Right, and our bodies are basically complex systems of fluids, right?
Exactly. Think about blood, lymph fluid, even the fluid around your brain and spinal cord. These are all fluids constantly moving around, carrying all sorts of things, nutrients, oxygen, waste products. So understanding fluid dynamics is crucial to understanding how our bodies work.
Okay, that makes sense. So where do we even start with such a complex topic? My notes mention osmosis.
Right, osmosis. It's the movement of water across a membrane, a barrier from an area where there's more water to an area where there's less. Think of it like water trying to dilute a stronger solution until both sides are equal.
Yeah, I remember that from biology. But how does it relate to our bodies?
It's all about balance. It helps keep the right amount of fluids inside and outside our cells. So, for example, if the fluid outside a cell has more stuff dissolved in it, like salts or sugars, than the fluid inside the cell.
Then the water moves out of the cell to try to balance things out.
Right, and that can cause the cell to shrink. But if the fluid outside has a lower concentration...
Then the water moves into the cell and makes it swell.
Exactly. And if that difference is too extreme, the cell could even burst.
Yikes. So it's all about maintaining that balance. What about blood pressure? How does that fit into all of this?
Blood pressure is the force of your blood pushing against the walls of your arteries. It depends on a bunch of things. How much blood your heart is pumping, how much resistance there is in your blood vessels, and how thick your blood is.
So it's like a plumbing system, where the pressure depends on the pump, the pipes, and the fluid flowing through.
That's a good analogy. And just like with plumbing, if the pressure is too high or too low, problems can arise. High blood pressure can damage your arteries, increasing the risk of things like heart attacks or strokes.
That's scary. The notes also talk about laminar and turbulent flow. What's the difference?
Laminar flow is smooth and streamlined. Picture like a river flowing calmly. Turbulent flow, on the other hand, is chaotic, all swirls and eddies. Think whitewater rapids.
So which one is happening in our blood vessels?
Most of the time, it's laminar flow, which is good. But sometimes, if an artery gets narrowed or the blood flow speeds up, it can become turbulent.
And I'm guessing turbulent flow isn't good.
Not really. Turbulent flow makes it harder for the blood to move through, meaning the heart has to work harder. This can contribute to high blood pressure and other heart issues.
It's amazing how all those factors, pressure, viscosity, the size of the blood vessels, all play a role in how the blood flows through our bodies.
It's a finely tuned system for sure. And, of course, the heart is the engine that keeps it all going.
Right. My notes go into detail about how the heart is structured and how it works, 4 chambers, valves, all that.
Right.
But what I really want to understand better is the cardiac cycle, that rhythmic beating of the heart. Can you explain it?
Sure. It's basically a sequence of events that repeats over and over. First, you have diastole, that's the relaxation phase. The heart chambers fill up with blood. Then comes systole, the contraction phase, when the ventricles pump the blood out to the body and the lungs.
Ah, that's where those lub-dub sounds come from, right? The valves closing.
Exactly. That first lub is the atrioventricular valves closing as the ventricles contract. And the second dub is the semilunar valves closing when the ventricles relax.
It's incredible that this cycle just keeps happening nonstop throughout our lives. But how much work is the heart actually doing?
Well, the work the heart does depends on how much blood it pumps and the pressure it needs to overcome. There's this thing called the Frank-Starling law that says, the more the heart muscle is stretched during filling, the stronger the contraction will be.
So it's like the more blood comes in, the harder it pushes it back out?
Exactly. It's a way for the heart to adjust to different conditions and make sure it's pumping out the same amount of blood it's taking in.
Pretty clever. So all this pumping and circulating, that takes energy, right? Which brings us to thermodynamics.
You got it. Thermodynamics is all about energy and how it changes. It might seem abstract, but its rules govern everything in the universe, from the smallest atom to, well, the beating of your heart.
I remember the laws of thermodynamics. Can you give me a quick refresher?
Sure. The first law says that energy can't be created or destroyed, only transformed from one form to another. It's like a cosmic bank account. You can move energy around, but you can't create it out of nothing or make it disappear.
So like that saying, you can't get something for nothing. What about the second law?
The second law introduces the idea of entropy. Entropy is a measure of disorder.
So a messy room has more entropy than a clean one.
Exactly. And the second law tells us that things naturally tend to get more disordered over time. It takes energy to create order out of chaos.
That's definitely true when it comes to my room. But how does this apply to our bodies?
Well, building complex things like proteins from smaller parts takes energy because you're creating order from disorder.
Okay, that makes sense. My notes connect thermodynamics to how proteins fold, which is something essential for life, right?
Absolutely. Proteins are long chains of amino acids that need to fold into specific 3D shapes to do their jobs. And that folding is guided by the laws of thermodynamics. The protein is trying to find its lowest energy state, which happens to be its correct shape.
The notes mention the Anfinsen experiment and something called Levinthal's paradox. What are those about?
Right, so the Amundsen experiment showed that a protein can fold itself into the right shape all on its own if the conditions are right. It's like the instructions for folding are built into the protein itself. But then Levinthal's paradox says that if a protein were to randomly try every possible shape until it found the right one, it would take longer than the age of the universe.
So how do they fold so quickly then?
Good question. That's where the idea of a folding funnel comes in. Imagine a funnel, and at the top is the unfolded protein, all jumbled up. As it starts to fold, it's like it's sliding down the funnel, getting closer and closer to its final, low-energy, properly folded state at the bottom.
So it's not randomly trying every shape. It's guided towards its correct form.
Exactly. And things like temperature, acidity, and even other molecules around the protein can influence this process.
It's amazing how we can use thermodynamics to explain something as complex as protein folding.
It really is. But let's switch gears for a bit and talk about another type of movement that happens in our bodies. Motor proteins. These tiny machines do all sorts of work inside our cells, right?
Right. They act like little delivery trucks, transporting things around the cell on these tracks made by cytoskeletal polymers.
Exactly.
And the notes mention 3 types of those polymers: microfilaments, microtubules, and intermediate filaments. Can you tell me more about them?
Sure. Microfilaments are made of a protein called actin. They form long, thin fibers and help give the cell its shape. They're also involved in things like cell movement and muscle contraction.
So they're like the cell's internal scaffolding.
That's a good way to think about it. Then there are microtubules. They're hollow tubes made of a protein called tubulin. They also help maintain the cell's shape, but they're also important during cell division. They form the spindle fibers that separate chromosomes, and they act like tracks for those motor proteins we were talking about.
So they're like highways inside the cell.
Exactly. And then we have intermediate filaments, which are made of a bunch of different proteins. They give the cell strength and help it resist stress.
So each type of these polymers has its own unique structure and function, but they all work together to keep the cell organized and working smoothly.
Exactly right.
And the motor proteins are what use these cellular highways to carry out all sorts of important tasks.
You got it. They're the little engines that make things happen.
The notes mention that motor proteins have these different parts: a head, a neck, and a tail. How do these parts work together to produce movement?
So the head is the part that actually binds to the track, the cytoskeletal polymer. It uses energy from a molecule called ATP to generate force and pull itself along the track. The neck acts like a lever, amplifying that force. And the tail is what attaches to the cargo the motor protein is carrying.
It's like a well-coordinated team. The head pulls, the neck leverages, and the tail carries.
Exactly. And this cycle of the head binding, pulling, and releasing, driven by ATP, is what makes the motor protein move along the track.
Fascinating. The notes mention some interesting terms: power stroke, working distance, stroke velocity, duty ratio, and processivity. Can you explain what those mean?
Sure. So the power stroke is basically that moment when the head of the motor protein pulls itself along the track, generating force. The working distance is the distance the motor protein moves during each power stroke.
So it's like the length of each step the motor protein takes.
Exactly. And the stroke velocity is how fast that step happens. The duty ratio is basically how much of the time the motor protein is actually bound to the track during each cycle. And processivity is a measure of how many steps a motor protein can take before it detaches from the track.
So some motor proteins are like marathon runners, able to travel long distances without stopping, while others are more like sprinters, taking short, quick steps.
Exactly. And all these properties are tailored to the specific job each motor protein has to do within the cell.
It's amazing how these tiny machines can convert chemical energy into movement. But speaking of movement, let's talk about muscle function. So the notes say skeletal muscles are made up of these bundles of muscle fibers, which contain even smaller units called myofibrils.
You're right. And inside each myofibril, you have these repeating units called sarcomeres. The sarcomere is the basic unit of muscle contraction.
And what's in a sarcomere?
They're made up of overlapping filaments of actin and myosin. And guess what? Myosin is also a motor protein.
So muscle contraction is basically just a really specialized version of what motor proteins are doing all the time inside cells.
You got it. And the sliding filament model explains how it works. The actin and myosin filaments slide past each other, which shortens the sarcomere, making the muscle contract.
Okay, I think I get that part. But the notes also mention these regulatory proteins called tropomyosin and troponin. What do?
They control when the muscle contracts. Think of it like a lock and key system. Tropomyosin normally blocks the sites on actin where myosin wants to bind so the muscle can't contract.
Okay, so it's locked.
Right. But when calcium ions bind to troponin, it changes shape and moves tropomyosin out of the way. So myosin can bind to actin, and boom, the muscle contracts.
So calcium is the key that unlocks muscle contraction.
You got it. And all of this is controlled by nerve impulses that trigger the release of calcium inside muscle cells.
The notes say the force of a muscle contraction actually depends on the length of the muscle. How does that work?
So the amount of force a muscle can generate depends on how much the actin and myosin filaments are overlapping in the sarcomere. There's kind of a sweet spot where the overlap is just right for maximum force.
So if the muscle is stretched too far or shortened too much, the overlap is less and the force is weaker.
Exactly. This is especially important for the heart. How much blood fills the heart stretches the muscle, which then influences how strongly it contracts.
It's like that Frank Starling law we were talking about. The more it's stretched, the stronger the contraction.
Exactly. It's a way for the heart to adjust to changes and pump efficiently.
Wow, the human body is truly amazing. But let's move on from muscles and talk about the mechanical properties of tissues in general.
Good idea. Understanding how tissues behave mechanically is important for figuring out how they work and how they respond to stress.
The notes use the example of a spring to explain this.
A spring is a great way to visualize elasticity. You push or pull on it, it changes shape, but then when you let go, it bounces back to its original form.
And the notes mention Hooke's law, which says that the force you need to stretch or compress a spring is proportional to how much you stretch or compress it.
Right. And that proportionality is called the spring constant. A stiffer spring has a higher spring constant.
So different tissues have different spring constants, right? Like bone is way stiffer than muscle.
Exactly. The stiffness of a tissue is measured by its elastic modulus.
My notes mention a bunch of different ways things can deform: compression, tension, shear, torsion, and bending. Can you give me some examples of what those mean for our bodies?
Sure. Compression is like when you jump and land, the cartilage in your knees gets compressed, squished. Tension is like when you stretch a muscle, it's being pulled. Shear is like when you rub your hands together, the forces are acting in opposite directions. Torsion is like twisting, like when you twist your ankle. And bending, well, that's like bending your elbow.
So our tissues are experiencing all these forces all the time as we move around.
Absolutely. And how a tissue responds depends on its properties. Some are more elastic, so they bounces back to their original shape, while others are more plastic, meaning they change shape permanently.
My notes also mention something called viscoelastic materials. What are those?
Viscoelastic materials have both viscous and elastic properties. Imagine something like silly putty. It can flow slowly, but also snap if you pull it quickly. They deform over time under constant load. This is called creep.
So they kind of remember their previous shape.
In a way, yes. And a lot of tissues in our bodies are viscoelastic. Cartilage, tendons, skin. It's what allows them to function the way they do.
It's amazing how all of these mechanical properties affect how our tissues work and how they get injured.
Definitely. But let's switch gears again. We've got another fascinating topic to cover. Have you ever heard of nuclear magnetic resonance, or NMR?
NMR. It rings a bell. I know it's used to study molecules, right?
Right. It's a really cool technique that uses the magnetic properties of atoms to give us information about the structure of molecules.
My notes mention the idea of spin. What does that mean in this context?
So in the world of quantum mechanics, atoms have this property called spin. Think of it like a tiny spinning top.
And just like a spinning top, they create their own tiny magnetic field.
Exactly. And when we put those atoms in a strong external magnetic field, their spins align with that field.
So they're all pointing in the same direction.
More or less, there are actually two ways they can align, either with the field or against it, and there's a tiny energy difference between those two states.
Okay, I'm following so far.
Now, this energy difference corresponds to a specific radio frequency. And if we hit those atoms with radio waves at that frequency, they absorb the energy and flip their spins.
That's the resonance part of NMR, right?
Yeah, got it. The atoms are resonating with the radio waves, and when they relax back to their lower energy state, they release radio waves, which we can then detect and analyze.
So what does this tell us about the molecule?
The frequency at which an atom resonates depends on its chemical environment. You know, what other atoms it's connected to and what kind of electrons are around. This difference in frequency is called the chemical shift.
So each type of atom has a unique chemical shift.
Pretty much. And that's what makes NMR so powerful. We can look at the chemical shifts in a sample and figure out what kinds of atoms are there and how they're connected. It's like a molecular fingerprint.
That's incredible. And the notes mention that NMR is used to study all sorts of things, small molecules, proteins, even DNA.
Yeah, it's super versatile, and it's used in all sorts of fields.
It's amazing how understanding these tiny magnetic properties can give us so much information about the world. But the notes also mention magnetic resonance imaging, or MRI. That's another technique that uses nuclear magnetic resonance, right?
That's right. MRI is basically a special kind of NMR that lets us create images of the inside of the body.
I'm definitely curious to learn more about that. Can you give me a quick overview of how MRI works?
Of course. So remember how we were talking about atoms having spin? Well, protons, which are the nuclei of hydrogen atoms, also have spin.
And since our bodies are mostly water, which is full of hydrogen atoms,
We're basically walking magnets.
that's a cool way to think about it.
So when you lie inside an MRI machine, a really powerful magnet aligns the spins of all those hydrogen protons in your body. Then radio waves are used to briefly knock those protons out of alignment.
And as they relax back into alignment, they emit radio waves that can be detected and turned into an image.
Right. Exactly. But here's the difference between MRI and regular NMR and MRI. We use these things called magnetic field gradients to spatially encode the signals.
Spatially encode, what does that mean?
It means we can figure out where those signals are coming from within the body, and that lets us build up a 3D image.
Oh, I get it. So that's how we can see all those detailed pictures of organs and tissues.
Right, and by analyzing those signals, we can also tell the difference between different tissues based on their properties, like how much water they contain.
That's amazing. The notes mention T1 and T2 weighted images. What's the difference?
T1 and T2 refer to how quickly those protons relax back to their equilibrium state after they've been excited by the radio waves.
So different tissues have different relaxation times, and that's what creates contrast in the MRI images.
You got it. T1 weighted images are good at highlighting tissues with a short T1 relaxation time, like fat. And T2 weighted images are good at highlighting tissues with a long T2 relaxation time, like water.
It's incredible how these subtle differences can be used to create such detailed anatomical images. No wonder MRI is so important in medicine.
It really is a game changer. And we can do even more with MRI these days. We can actually see activity in the brain, for example.
Wow, that's incredible. It's amazing how far we've come in understanding the microscopic world and using it to improve our lives.
It really is remarkable.
Well, I think it's time for us to shift gears and explore another fascinating part of the microscopic world, the sense of hearing.
Sounds good to me. Hearing is a complex process that involves converting sound waves into electrical signals that our brains can understand.
Okay, I'm ready to dive into the world of acoustics. Okay, so how does hearing actually work? I know it all starts with sound waves hitting our ears, but then what?
Well, those sound waves are just vibrations in the air, right? They travel through the ear canal and hit the eardrum, making it vibrate too.
So it's like a drum head.
Exactly. And those vibrations from the eardrum get passed along to the middle ear.
Okay, the middle ear.
Right. And then there are these three tiny bones, the malleus, inticus, and stapes. They're like a mini lever system, amplifying those vibrations and sending them to the inner ear.
Wow, I remember seeing those tiny bones in diagrams. It's amazing how such small things are so important for hearing.
It really is. The middle ear also helps to make sure the pressure inside the ear is the same as the pressure outside.
Oh, right. That's why our ears pop when we're on a plane or going underwater.
Exactly. It's the eustachian tube that connects the middle ear to the back of your throat that does that. But the really cool stuff happens in the inner ear.
Okay, the inner ear. What's going on in there?
That's where the cochlea. It's this spiral-shaped organ, and it's filled with fluid.
Okay, the cochlea.
Right. And when those vibrations from the middle ear reach the cochlea, they create waves in this fluid.
So it's like a little ocean inside my ear. But how do those waves turn into the sounds I hear?
Inside the cochlea is something called the basilar membrane. Imagine it like a ribbon that runs along the length of the cochlea. And get this, it's tonotopically organized.
Tonotopically, what does that mean?
It means different parts of the membrane vibrate depending on the frequency of the sound.
Oh, I see. So high-pitched sounds would make one part of the membrane vibrate, and low-pitched sounds would make a different part vibrate.
Exactly. And on top of the basilar membrane, there's the organ of Corti, which is where these special cells called hair cells are.
Hair cells, what are those like?
They have these tiny hairs called stereocilia that stick out into the fluid. When the basilar membrane vibrates, it makes those stereocilia bend.
And bending the hairs, that's what creates the signal for sound.
You got it. When the stereocilia bend, it opens up ion channels in the hair cells. That changes the electrical charge of the cell.
Okay.
And that change triggers the release of neurotransmitters.
Neurotransmitters, those are the chemicals that brain cells use to communicate, right?
Right. And those neurotransmitters activate the auditory nerve, which then sends signals to the brain.
Wow, that's a lot of steps.
I know, right? It's a pretty complex process. But in the end, those vibrations in the air get turned into electrical signals that our brains can interpret as sound.
It's amazing how the body can do all that.
It really is. It shows how sophisticated our sensory systems are. Speaking of sound, maybe we should talk about a technology that uses sound waves in a different way, ultrasound.
Oh yeah, ultrasound.
Mm.
I know it's used a lot for imaging, like when someone's pregnant.
Mhm.
But how does that work? Does it use radiation, like X-rays?
Actually, ultrasound is totally different from X-rays. It uses high-frequency sound waves to create images, so it's super safe and doesn't use any radiation.
So it's like a really powerful version of sonar.
That's a good way to think about it. So an ultrasound machine has a transducer that sends out pulses of these high-frequency sound waves. The waves go through the body and bounce back like an echo when they hit different tissues.
Okay, like an echo.
And by measuring how long it takes for those echoes to come back, the machine can figure out how far away the tissue is and create an image.
But how could it tell the difference between different types of tissue?
Well, different tissues have something called acoustic impedance, which basically means they reflect sound waves differently. Some tissues reflect more sound than others, and that creates contrast in the image.
So that's why bones look different from organs on an ultrasound image.
Exactly. And by analyzing the strength and timing of those echoes, the ultrasound machine can create detailed images of organs and tissues inside the body.
That makes sense. The notes also mention the Doppler effect. Doesn't that have something to do with how the pitch of a sound changes as something moves towards or away from you, like a siren getting louder as it approaches?
You're exactly right. Doppler ultrasound is a special type of ultrasound that's used to measure blood flow. See, when sound waves bounce off moving red blood cells, their frequency changes a little bit, just like the pitch of that siren. And by analyzing that change in frequency, we can figure out how fast the blood is flowing and in what direction.
So it's like a tiny radar gun for your blood cells.
Exactly. And that's super useful for finding blockages in arteries, checking blood flow in organs, and even monitoring a baby's health during pregnancy.
Wow, it's amazing how we can use sound waves for so many things.
It really is. And I think it shows just how versatile and powerful physics is, you know? It helps us understand and interact with the world around us, from the tiniest atom to our own bodies.
Absolutely. We've covered a lot of ground in this deep dive, from atoms and light to cells and senses.
It's been quite a journey. Hopefully it's given you a new appreciation for all the invisible forces that shape our lives.
It definitely has. I feel like I've learned so much about how things work at the microscopic level.
It's been a pleasure exploring these topics with you.
A huge thanks to our expert for guiding us through this amazing world. And to our listener, thanks for joining us on this deep dive. Keep exploring and asking questions. Until next time.